All right, it looks like almost everyone has joined, so we will start off. Hi, everyone. Thank you so much for joining our webinar today. My name is Julia Saracini, and I'm a member of the ACER consulting team that is hosting this event series today in collaboration with the Dairy Farmers of Canada. Today is our 12th and final session, actually, in this series that we've been doing since September, and this series has been highlighting dairy research and dairy research projects from across Canada. If you've been joining us for these webinars over the last 10 months, thank you so much for your participation and your continued support. It's been great to have you see a lot of familiar faces every few weeks, and we really appreciate the participation. For today's session, our focus is going to be on designing a reference population to accelerate genetic gains for novel traits in Canadian Holsteins. And I would like to remind everyone that, as with our previous sessions, we are hosting this event as a traditional Zoom meeting, which means that you have the ability to turn on your cameras and microphones. However, for the best audio quality and participation, we're going to ask everyone to please keep yourself muted until you've been called upon for a question or until you have a comment for our speakers today. We will be taking questions in the chat that we'll be answering at the Q&A session towards the end of the seminar. So at any point, please feel free to send your questions to myself, Julia Saracini, or my colleague, Carly Wright, who's identified as the host for this meeting today. And we will do our best to address all questions in the Q&A period towards the end. Our format of today is going to be presentations for about the next 40 minutes, and then a question and answer period that we would encourage you all to participate in towards the end. If you would like to hear this event in French today, you can access the French interpretation feature through Zoom by clicking on the interpretation globe at the bottom of your screen, selecting the French language option, and then muting the original audio. We do have live interpreters today who will be interpreting the session in French for you. If you need any assistance throughout this event with any technical features, or if you have any questions, please feel free to contact myself or Carly Wright in the chat function at the bottom of your screen. I'd also like to remind everyone that the event today will go until 2:00 p.m. Eastern, and we will be having individual researcher presentations followed by our question and answer session. I'd now like to hand things over to Amy Desilets to provide acknowledgements from the Dairy Farmers of Canada.
Thank you, Julia, for the introduction. My name is Amy Desilets. I'm Assistant Director at Dairy Farmers of Canada, Assistant Director Research. I just want to thank you all for registering for today's webinar. This webinar series has been a success. It was launched by the FC last fall as part of our ongoing commitment to research communication and knowledge translation. This webinar series aimed at showcasing really all the exciting work that is going on in dairy research across Canada. This initiative was possible thanks to the financial support of DFC, but as well as our partners. And I'd also want to thank all the research team from across the country for their collaboration with ACER for all those events. And final thanks maybe to ACER as well for essentially organizing and coordinating everything for those events. So without further ado, I'll now turn the floor over back to you, Julia.
Thank you, Amy. I'd now like to introduce our first of 4 speakers for the day. So we're going to have our introduction by Dr. Flavio Schenkel, who is the primary investigator for this research project. Flavio is a full professor with research interests ranging from theoretical to applied genetics and genomics in livestock breeding. His current research focuses on the use of genomic information to enhance genetic evaluation of livestock species with an emphasis on genomic selection. Since 2013, Dr. Schenkel has served as the director of the Center for Genetic Improvement of Livestock at the University of Guelph. So I will hand it over to you, Flavio.
Okay, thank you, Julia, for the introduction. And good afternoon, everyone. I'd like to thank you very much for attending our virtual research showcase. We are really pleased to present to you a sample of the investigations that we carried on as part of this large dairy research cluster 3 project that was entitled Designing a Reference Population to Accelerate Genetic Gains for novel traits in Canadian Holstein. As I mentioned, my name is Flavio Schenkel, and I was the principal investigator of this project here in our Department of Animal Biosciences at the University of Guelph. But I like to acknowledge that initially, this project was led by Dr. Filippo Miglior, that had to leave the project very early, a few months into the project, and then I took over as the principal investigator. So the main goals of this large project were, can you pass this slide, please? The first main goal was to design a cow reference population for future research, but also for the development and implementation of genomic selection for novel traits. That was the goal. And why a reference population for novel traits? Because those are traits difficult to measure, where you have a limited number of phenotypes, and genomics could benefit much more. So we end up with the project genotyping about 26,000 cows that were recorded for novel traits, not for all novel traits, but most of them for at least one novel trait. The second objective was to develop genomic evaluation for these various novel traits using what we call single-step methodology. For instance, of novel traits here, I could list feed efficiency, methane efficiency, heat tolerance, among others. And the third goal was to perform genome-wide association analysis, functional analysis, and fine mapping of genes for these novel traits, so that you would know better the genetic architecture of the novel traits. But today, because the scope of this project was quite broad and a lot of work was done, we are going to focus on 3 studies as a sample of what was done. And these 3 studies, they focus more on novel methodologies and traits. Next slide, please. So the presenters today will be, the first one will be Dr. Luca Alcantara, who finished his PhD program last year. And Dr. Alcantara is currently the Research Center Data Manager at the University of Guelph. And Dr. Alcantara will present one example of use of artificial intelligence applied to cow fertility evaluation. The second presenter will be Ivan Campos. Ivan is a former master student with us, but also now is a current PhD student. And Ivan will summarize our studies aiming to implement genetic evaluation for heat tolerance in Holstein cows. And finally, as the last presenter, will be Dr. Saeed Shadpour. Dr. Shadpour is a postdoctoral fellow in our research group, and he will present a recent study on predicted methane production using milk mid-infrared spectrum. So I hope that these 3 examples are going to give idea of the scope of the project and the success of the project. And I hope everybody is going to enjoy our showcase. And I will pass then the microphone, in this case, to Dr. Alcantara to present the first part of the presentation, the first set of investigation. Thank you very much and enjoy.
All right, well, hello everyone. Thank you, Flavio, for your kind introduction. Today we are discussing about the impacts of artificial intelligence on genetic evaluations in the dairy industry, more specifically on fertility traits. The work that I'm presenting here is from my PhD thesis when I was under the supervision of Dr. Schenkel and Dr. Bass, and we published this on the Journal of Dairy Science last year. So Timed artificial insemination protocols is a successful management tool in dairy cattle. It affects the reproductive physiology and endocrinology involved in the estrus cycle, and it allows for higher conception rates even for genetically inferior animals in terms of fertility. However, timed AI is known to mask an animal's true fertility performance, as it reduces the accuracy of genetic evaluations for fertility traits, as recently reported by Oliveira and Lynch. Next, please. One of the suggestions proposed by Lynch to address this possible bias was to split fertility traits by the management technique involved in the breeding. The goal with this is to have unbiased genetic evaluations of animals according to their true fertility performance. Next. But this task is rather difficult because there's a lack of specificity and uniformity in the recording of breeding protocol descriptions by dairy farmers. This makes it very hard to identify which protocol descriptions are from timed AI protocols and which are not. Next. But what are these protocol descriptions anyways? When an animal is bred, a large number of farmers indicate on dairy comp the breeding protocol code used on that service. Each breeding code is a letter that is linked to a short description of that breeding protocol. And that is normally a combination of only a few keywords, abbreviations, numbers, or special characters such as natural, off-sync, standing, GGPG, and cider. However, next, because there is no standard for creating these protocol descriptions, two farmers might use different descriptions for the same or variations of the same protocol. For instance, Lynch reported that there were 156 unique descriptions just for off-sync in their data set. And the source of variation came from typos, capitalizations, and or abbreviations, as you can see here, some examples at the bottom of the slide. Next. Another factor that makes working with breeding data challenging is that farmers create some descriptions that are very much not clear, or for protocols that are hormonal treatments, not necessarily for timed AI, but for sick animals. So, for example, we have 15 plus hours or just astromate or astroplant. So not very clear. Next. So the data set Lynch used from Canadian farms contained almost 6,000 different breeding protocol descriptions across around 1,000 herds, of which 2,000 of them were herd specific. And by the way, this word cloud that you see here was created from his data set. So that's an actual word cloud. Next. So we use the same data set from Lynch, and it contained breeding data recorded by Canadian Holstein farmers on dairy comp from 2007 to 2019. There was data from almost 800,000 cows spread among 1,100 herds. We removed herds that were not recording the breeding protocols that they used, and we were still left with records from around 70% of Holstein cows in Canada, which gave us a great representative sample at the end. Next. We leveraged Lynch's previous manual classification, and we split the protocols into two classes. So timed AI, number one, when timed AI protocol was used alone, like just off-sync, or in combination with any other protocol, like PRID and astromate or off-sync and natural. And the second class was other, when the protocol description did not indicate any use of timed AI protocol, just, for example, standing heat or astromate or even vet advice. So we assumed that Lynch's classification was highly accurate when he did that. Next one. So next thing we did, we looked at the number of new unique breeding protocol descriptions added to the data set every year. We can see that the number of new codes being added only stabilized after 2010. We also noticed that there was an average addition of 140 new codes every year after that, so starting from 2011. 140 new codes every year. So we understood that in that moment that we needed to build a model that would be robust enough to maintain a high performance, even when with this number of unseen data being added every year. So in other words, we needed a model that could easily classify breeding records into timed AI or not timed AI without frequent training. But how can artificial intelligence, or more specifically in this case, machine learning, can help the dairy industry deal with these different protocol descriptions and allow for an unbiased genetic evaluations of fertility traits? So let's take a look at what we did in this work. Next. So our first approach was to convert those text-based protocol descriptions into a table that a machine learning model can understand. So the first thing was we cleaned those descriptions to ensure that all protocol descriptions were lowercase and had no underscores. Second, we extracted unique bigrams of 2 consecutive characters from each protocol, and then we created a dictionary for each data set. So timed AI had TIMMEEDAI as an example. Our last step was to convert protocol descriptions into table that contain frequency counts of those 2 characters. And then we use these tables to train the models. Next. Our next approach was to mimic what would happen in the future. So we trained the models with only a certain amount of data up until a certain year and tested it on each of the following years. So for example, in the first scenario, first row you see there, we trained the model with data from 2007 to 2010 and tested to see how that model performed when it saw the breeding protocols from 2011 for the first time. And then we did that for 2012 and then 2013 and so on. Next, we tested to see if adding the newly created codes from 2011 helped predict which timed AI protocols were used in 2012, 2013, and so on and so forth. So we kept adding 1 year at a time and evaluating on the following years until there was only 1 year left to test. So a larger training set and a smaller, just 1 year to test. Next. So after optimization and model hyperparameters, optimization, the training with a stratified 10-fold cross-validation approach, we tested our models on unseen data, and these plots show the results we obtained from the 8 different models we trained. On the x-axis, or the horizontal axis, on these plots, it shows the training sets. So each of those orange boxes represents the results from models tested with a different amount of data. The performance of these models on unseen data is shown as F1 score on the Y or vertical axis. If you're not familiar with F1 scores, this is a measure of performance that the closer to 1, the better. So in our study, a perfect score of 1 means that all timed AI observations were correctly classified as timed AI, and all observations classified as timed AI are truly timed AI. So the orange boxes on the distribution on the F1 scores or the results of a particular model on unseen data. So on the first box, we have the results of a decision tree model trained with data from 2007 to 2010 and tested on each of the following years, 2011, 12, and so on until 2019. At the end of this plot, the last box is actually a solid line because the model was trained with data only from, 2007 to 2018, and it was only tested on the 2019 data. So as you can see from the fluctuating pattern within these blocks, we can see that training these models with more data every year did not help increase F1 scores, as we see the boxes within each plot are pretty much at the same height, except for models based on linear discriminant analysis. This shows us that the machine learning algorithms used in this study can be used to produce models with consistent high F1 scores, regardless of the amount of data used for training. And this is the great news because we don't need to retrain models frequently. You can also notice that the average F1 scores are very similar for all models. And this is also good because it gives us a better flexibility when considering and choosing an algorithm to do this kind of job. So, okay, so from the results of this work, we now know that artificial intelligence can be used to correctly identify which breeding protocols use timed AI to inseminate Holstein cows. It can positively impact the dairy industry by opening the possibility for unbiased genetic evaluation of animals based on their natural fertility. And it can also help select for cows that perform better under a specific type of protocol, or just to help for bulls that have daughters with superior performance for natural and or timed AI fertility. So next. With that, I'd like to thank you for listening, and I'm happy to take any questions at the end. Thank you.
Thanks, Lucas. As Lucas mentioned, he will be taking some questions towards the end. So if you have any questions, please feel free to put them in the chat and we will address those towards the end. I'd now like to welcome Ivan to present his research and take it away.
All right. Can you all hear me?
Yes,
I hope
thank you.
so. Thank you. So my name is Ivan. I'm also a student under the supervision of Dr. Schenkel, and today I'll be sharing with you my research, which has to do with heat stress and how that has impacted the dairy industry in Canada. And so the title of my presentation is Developing Genetic and Genomic Models to Assess Heat Stress Resilience in Dairy Cattle. So next, please. So to start us off, I believe it's important for us to, I guess, remember at this point that heat stress is an issue for the Canadian dairy industry. So as you can see, the average temperature has increased over the years. And not only that, but we are actually bombarded with new reports every year. In the news, we also see things talking about climate change and global warming. And even in a couple of into 2021, I believe there is a heat wave hitting BC, and that was quite drastic. And we saw that impacting people. We can think of that also impacting the animals. So next slide, please. So I believe that if you want to look and understand a little bit more of the impact of heat stress, we may start with a simple graphic of a thermoregulatory profile of mammals. Next slide, please. So as you can see there, here's a simple version of this thermoregulatory profile, and we can think of a mammal like a cow. And we look at the thermal comfort zone. So there is a range of temperature that the animal will spend the least amount of energy towards thermoregulation. And that's I would say, the comfort zone. Actually, that's the name of it, thermal comfort zone. But now if we go with the temperature above or below some critical temperatures, please next, and then next again, we see that the animal will start using physiological mechanisms to thermoregulate. And therefore, energy will be now spent towards this physiological mechanisms. Next, please. So in my case, as long as we are talking about heat stress, we want to look at this side of the graphic where we have the upper critical temperature. So as temperature goes up this or above this critical temperature, the animal enters heat stress. Next, please. And then, as I said, energy is shifted towards thermoregulation. So if the animal is producing milk, now less energy is directed to production, and it goes towards thermoregulation. And if the temperature continues to go up, next please, we get to a point of severe heat stress. So why heat stress is important? I believe there are many consequences that we may be aware of, but I would summarize into a few points. And one important thing is that when we are thinking of heat stress, the animal needs to mainly or mostly dissipate heat, but also stop some actions that would produce heat itself. So one of the particular aspects of an animal under heat stress is that the animal will stop eating. Next, please. And so that will result in a reduced feed intake. So this is a short-term result that we observe when animals are under heat stress. But there are also other implications. And next, please. Other studies, they looked into reproductive performance, and they have found that there is a negative effect due to heat stress. And so for us, what we started looking into considering our studies was, in fact, production. So next, please. And we identified that there is a relationship between temperature and production. And in fact, what happens is as temperature goes high and above the upper critical temperature, milk yield starts to decrease. So next, please. And so here I show you a real data from Canadian farms where we have protein yield and the temperature humidity index. It's an index that combines temperature and humidity. So as you can see here, this relationship shows us that to a certain THI, or the temperature humidity value, protein yield starts to decrease. Next, please. And that's what we found for protein yield, but we also found for fat yield. Next, please. And the same thing we saw, I think one more, please. Yeah, so here even more easy to see this relationship. And for fat yield, we have that at certain temperature humidity index, we see the production decline. Next, please. So all that actually translated into a economic loss. So we have done this in our study and estimated roughly what those productions losses would be translated in economic losses. And we found that it's about $30 million per year to the Canadian dairy industry. And the next thing that I would like to point, considering the effect of heat stress, is in the next slide, which is talking about how high-producing cows, they are generally the ones that suffer more with the temperature. And that's why, that's the reason why, is because they have so much energy input towards production, and there's just a small room left for thermoregulation. And it's important to consider, so Canada now, for its high producing cows, that needs to consider this additional importance when thinking about heat stress. So next slide, please. So let's start thinking about the animal and the heat exchange between the animal and the environment, because that is very important to understand a bit more of how heat stress impacts our dairy industry. So there are factors involving in this or influencing this heat exchange. So the first one we can say is the environmental factors. So please, next, yes. And then we can think of temperature and humidity, as I mentioned, but there are other factors, and the next is microclimates, and we can think of, next slide, please. The stocking density and the type of barn, those are examples of things that will interfere, will influence the heat exchange between the animal and the environment. And lastly, there's also animals' characteristics, and we can think of genetics, age, and if it's a lactating cow or non-lactating cow. And next slide, I focused my studies on genetics. So in the next slide, thank you. There are different ways that we can tackle heat stress, and that involves different fronts. For example, we can think of implementing heat abatement devices, and the next slide. And that would allow us and allow the producer to control the environment and control temperatures for where the animals are. That will prevent those animals entering or experiencing heat stress. But we also can work with genetics. And the next slide, please. It involves genetic information combined with performance, and that test day records is what we used. So genetic information, they come usually in form of a family relationship. And next, we can see a pedigree there, but also DNA, so genotyped animals. And with these informations combined, what we can do, we can actually perform genetic evaluations and identify animals based on their genetic merit for production, but also for heat tolerance. That's in the next slide. And so for that studies, the next slide tell us that we need environmental variables, and that involves ambient temperature, relative humidity, and the index, as I mentioned before, that combines these two environmental variables. And next, please. We started using information publicly available from weather stations. So we looked at the closest weather stations to all herds we had in our data set, considering Canada, to collect this environmental information. So what happened though is that weather stations, they are not so consistent in terms of the records, and depending on the time frame that we want to look, considering our test day records, the amount of data that we have, we may get some limitations there, which in fact resulted in us being able to work with a few herds, enough herds for our initial study. But then we had another idea that was to explore another source of environmental variation, or not variation, variables, sorry, it's in the next slide, which is the NASA powered database. And that allowed us actually to work with as many herds as we had available with the performance records, because it combines different sources of environmental variables, and it includes in this database from NASA, and that is pretty much for the entire world. So we can select the location of the farms, and we have this information. Next slide, please. So in our studies in Canada, we have identified this, there is a genetic variability considering the capacity of the animals to cope with heat. And that is important because that allows us to work with genetics, and we can select animals that are more heat tolerant, and through breeding, we can pass on their genetics, and therefore we are providing here a solution that is cumulative and permanent, and that will improve the overall heat tolerance of the Canadian herd. Next, please. So in my PhD, and actually I started in my master's, me and Flavio, we looked at this way to model heat stress and include when thinking of estimating breeding values. So as I showed, we had information from environmental information and test day records, and we came up with a model that would take into account not only production, but heat tolerance. Next, please. So our goal, considering climate change and what Canada will face, considering all this global warming, is to provide to the producer a range of animals that are not only high producing animals, but they also cope well with heat. And so the next slide, please. Throughout the years, we have came up with a total of 4 articles published. And if you want to know more about it, you can go, and it's publicly available as well. And next, please. So as conclusion of this presentation, heat stress, we are aware it's a problem here in Canada. It affects the dairy industry, and through genetics, among other potential solutions or ways to tackle this challenge, we can improve the thermal tolerance of our animals, our cows. And that's what we've been doing in our research here at the University of Guelph. So thank you very much. The next slide is the questions, which I'll be happy to take after the next presentation.
Thank you, Ivan. Yes, we will be taking questions, as I mentioned, towards the end. We've had a few come in, so please keep those coming. And next, we will welcome Saeed to the floor.
Thank you, Ivan. Hello, everyone. My name is Saeed Shadpour, and today I would like to use this opportunity to give you an overview of my research project on the estimation of genetic parameter of the predicted methane emission in Canadian Holstein dairy cattle using random regression model, supervised by professors Christine Bass, Dan Tulpan, and Flavio Schenkel. Next slide, please. First, let's see if mitigating methane emission is really important. Methane emission has 32 times more global warming effect than carbon dioxide. Methane emission is also responsible for 52% of greenhouse gas emission in the world. Reactive methane emission is responsible for 18% of anthropogenic greenhouse gas emission, and it is also responsible for 3 to 10% loss of the total energy intake. So by mitigating methane emission of dairy cattle, its environmental footprint will be decreased, and at the same time, feed efficiency of dairy cattle will be improved. Next slide, please. Measuring methane emission is expensive and labor-intensive, so small data sets are available, which limits the genetic improvement of the trait. The prediction of methane emission using a prediction model such as artificial neural network model is a good solution that make it possible to have millions of records. Next, please. We know that first, the production of fatty acids in the rumen is proportional to the erected methane emission. Second, the production of fatty acids in the rumen is also proportional to the fatty acids in milk. And finally, we know that milk-meal data is a highly accurate predictor of milk fatty acids. So we can expect that milk-meal data can be applied to predict methane emission in dairy cattle. The two major benefits of predicting methane emission using milk-meal data are obtaining a large number of predicted methane emission records, which is required to increase the accuracy of selection, and prediction methane emission using milk-meal data does not involve any additional costs, since milk-meal data already have been collected in commercial farms at a reasonable cost. Next, please. We applied artificial neural networks model to predict methane production. Prediction using artificial neural networks does not require a predefined model. Actually, artificial neural networks can learn from the data. Artificial neural networks have a black box behavior, which means that it is not easy to understand what is the real relationship between inputs and output of the networks. But this property does not matter in most applications. Complicated artificial neural networks are computationally demanding, but it is not true for simpler artificial neural networks like the ones used in the current study. Next slide, please. The calibration data to make prediction models consisted of 458 prediction methane production records from 426 cows across 2 herds with days in milk ranging from 5 to 229. We built one linear artificial neural network model and 10 nonlinear artificial neural networks model, where the number of neurons in the hidden layer of the nonlinear neural networks model was changed from 1 to 10. The nonlinear neural network model with 2 neurons in the hidden layer provided the best prediction accuracy equal to 0.65. The mere data and season of calving were used as predictor variables to predict weekly average methane production. Next slide, please. After finding the best prediction model, the model was used to predict methane production record. Before estimating the genetic parameter using the random regression model, some filters were applied to control the quality of the predicted methane production record and reduce the size of the data. The final analyzed data consisted of more than 30,000 records from more than 6,000 cows distributed over 250 herds. Before applying the random regression model to estimate the genetic parameter of predicted methane production, we applied a repeatability model to get preliminary results. The data that was analyzed by a repeatability model belonged to the middle of lactation. It's worth mentioning that the result of the repeatability model urged the CDN to start some measures to include methane production in the selection index. To get a better understanding of genetic parameter of predicted methane production over lactation, random regression model was applied later. The result of random regression model were in line with the result of the repeatability model. Today, I will present the result of random regression model. Next slide, please. Here we have the estimated heritability of the weekly average predicted methane production. The estimated heritability decreased from the first week of lactation to the 5th week of lactation, and then almost linearly increased over the lactation. The average estimated heritability of the predicted methane production was about 40%, which is a moderate heritability. Next slide, please. Here we have the estimated genetic correlation between methane production and production trait. The estimated genetic correlation between methane production and fat yield varied over the lactation with average 0.20. The estimated genetic correlation between methane production and both milk and protein yields varied over the lactation and were negative with average of minus 0.26 and minus 0.17 respectively. Next slide, please. Based on the results of this research, we can conclude that genetic parameter of mere predicted methane production changed substantially from early lactation to after the peak of lactation. Heritability decreases in early week of lactation, increasing after that. Genetic correlation with production trait change more in the early lactation, stabilizing more after that. Overall, mere predicted methane production is moderately heritable and can be used for genetic selection to mitigate methane emissions in dairy cattle. Next slide, please. Overall, the estimated negative genetic correlation between mere predicted methane production and both milk and protein yields suggests that increased milk and protein yields are associated with lower methane production. Overall, the positive genetic correlation between mere predicted methane production and fat yield suggests that increased fat yield is associated with higher methane production. The unfavorable genetic correlation with fat yield should be considered when selecting for lower methane mere predicted methane production to avoid potential negative impact on the fat yield. Next slide. In the end, we would like to acknowledge all the funding agencies and project partners who made the dairy cluster's research work possible and would be happy to answer the questions. Thank you very much for your attention.
Thank you, Saeed. So we are going to open up for a question and answer period now with our presenters. So if you do have a question at this time, feel free to put it into the chat. We have a few questions that have come in already. So the first is a question for Lucas. Lucas, was analysis done in the Canadian Genetic Evaluation for fertility, adding a correction for the use of timed AI? If yes, how did the results change in comparison to a model without taking that fact into account?
Hi, yes, that's a great question. No, we haven't done that yet. So that is one area that we are interested in doing and comparing from what we got from the model and from without the model. So we haven't done that yet.
May I jump in?
Of
Sure.
course.
Yeah, no, Lucas, I think the question was more general, was for the group. This was done before, like Lynch did exactly this.
Okay.
Compare the model accounting or not accounting, and there was important re-ranking of the sires. So and also the trade. If you consider like timed AI, the fertility under timed AI and fertility without timed AI, they are not exactly the same trait genetically. So the correlation is not close to one. And but prior to Lynch, there was a study by Oliveira that did a simulation study without real data, but simulated this situation or have a cows under timed AI, cows the natural, no timed AI, but in fact was timed AI, no timed AI. And he showed that there was not only re-ranking, but also the selection response would change. So we have a less selection response if you don't separate the two types of observations. So that's what simulation. Then we did with the real data and confirmed re-ranking. So, but Lucas didn't do like
Sorry, I understood. I understood if we had run the genetic evaluations again with the traits split through the model that we created. That's what I got.
Yes.
So no, we did it. Lynch did it, yes, but not us. Yeah. And there was a significant re-ranking of bulls depending on the records that were used. Yes.
Thank you. Another question for Lucas about your machine learning algorithms. So wondering if the algorithm can read text-based information from herd management software that's been recorded in English, but also in French.
Yes. So the general idea of this model is that it would convert text into frequency counts, and it would get the pattern of those frequency counts and then classify and estimate those. So the fact that most of our data set was in English, that doesn't mean that other languages, it wouldn't apply as well. So if there is a high amount of data with other languages, probably we will have to retrain that. But given that the protocols used for timed AI are very similar, for example, in English and French, I don't expect to see much of a difference between the two. But yes, it's possible.
Great, thank you. We do have a couple questions for Ivan. So Ivan, is genetic variation in heat stress among dairy cows correlated with any other production traits? For example, heat stress tolerance correlated with high producing cows. You mentioned that there is eventually going to be an opportune time to look for cows that kind of have both of these traits, high producing, but also heat tolerance. Can you speak a little bit about that in terms of any associations seen between those preferable dairy producing traits, but also the importance of heat tolerance?
Yeah, sure. So I'll start off with something that I've done, looking specifically considering production. So the way we actually evaluated heat tolerance was based on the decline of milk, considering that particular temperature humidity index. And therefore, what we looked was a phenotype was how much of there's a decline when the temperature exceeds a certain threshold. And with this, considering that, we looked and we found a negative correlation between that decline, which would be telling us the tolerance, okay, towards heat with production. So the slope is negative correlated with production, which tell us that there is, in fact, cows that generally producing more, they are generally the ones suffering more. However, the correlation was not a unity, which also tell us that, in fact, we could consider both production and heat tolerance in our evaluations. So we are actually studying models to really give us accurate results that can be also validated with other traits. I have not looked into different traits, for example, fertility or other novel traits. I've not looked with the correlation between heat tolerance for now. In the literature, they are already doing it, so we can find results showing, particularly for reproductive performance, so fertility traits, there is this effect that is transgenerational as well. So that's the first part of the question, I guess. So yes, there is a genetic correlation and it's negative considering production and heat tolerance, but it's not unit, which means that there is a room for animals that are high producers, but they also cope well with heat. And now the question is how we can implement an index. So we are still looking at different ways that we could work with those traits together to have them implemented for selection. I don't know if Flavio wanted to add something else to this question.
No, if I just would add that it's not unit, and in fact, it's about 0.3, the correlation. So it's not very strong correlation, but the correlation is there. And so showing that more the cow produce, she's going to be more sensitive to the heat stress. But it's not, because this correlation is not perfect. There are for sure cows that produce well and are more tolerant. And that's where the cows that we are looking for. In fact, we should look at this at the sire level, right? We are going to have a sire that produce cows that are highly producing cows, but heat tolerant. And we can see this, we see in our data set. But I just like to point the correlation about 0.3, 0.30, right?
Thank you. Another question has come in for Ivan, so I'll just tack that one on as well. Wondering if you saw an important re-ranking of BV values for sires when you were considering heat tolerance, and about how accurate that was.
Okay, yeah, those are good questions that I may not have from the top of my head the numbers, but in terms of re-ranking of breeding values, yes, we saw, and it was particularly for the top 10 animals that we had in our data set. And I can send information on that we have published and I have presented in a field conference where we actually have the breeding value of animals over or under thermal comfort. And then after a certain temperature humidity index value and how that breeding value decreased and the re-ranking of animals. So we have looked into that, and there is a considerate re-ranking of animals, especially among those top sires. That would tell us a little bit of, continue this evaluation, these studies. It's really important to really find those animals that can cope well with, produce well and cope well under heat stress. Yeah.
I just like to add something to, and I think it's important to say that this re-ranking is especially more important for fat and protein, especially fat. And those are the components that the producer paid for. For milk production, the less re-ranking, but for fat and protein, much more re-ranking. And a point that we could even genetically tell that there is a genotype by environmental interaction. So it means that that's a re-ranking or rank correlation below 0.8. So that's important. And why milk is different? Because it's interesting that milk at certain point when the cow gets stressed, even increase the production at the very beginning. Because the cow start drinking more water, right? And so it affects even the milk production increasing. But at a certain point, just drinking water doesn't do, to control the temperature. So and then the milk start decreasing. But fat and protein, the solid, they start to decrease much earlier in terms of the temperature and humidity. So those are important aspects. But yes, the answer, yes, there is re-ranking. So again, showing the opportunity that we could select for sires or bulls that are, the daughters produce well, but also are tolerant to heat. Because again, we see this variation, right, in the data set.
Thank you. We do have one last question for Ivan and then a couple for Saeed that have come in. So Ivan, the last question here is asking if you're aware of any country globally that has developed national genetic evaluations for heat tolerance in dairy cattle. And if there are any countries, how important should this trait be in Canada? How worried should we be about heat tolerance, given that we are a northern country? And what are some of the things that other countries have been developing to do some of these genetic evaluations regarding heat tolerance for dairy?
Yeah, thank you, Julia. That's a very good question that actually captures everything, I guess, that my research is trying to come up with answers for. So the first question related to other countries and how this trait has been looked upon. So I know Australia has already implemented a genomic breeding value for heat tolerance. In fact, it is interesting that in the 2000s, the early 2000s, a group from the United States, they started looking at how to model heat tolerance. And they were, I believe, the precursors of this type of studies when we could select animals based on their genetic merit for heat tolerance. And then the Australians, they went further, they worked with genomics, and they implemented, they could estimate the variance components, all necessary for the implementation of this trait, which is now in used by their program. So producers there, they can find a rank that takes into account that the heat tolerance, right? There is, or there are, but I will mention one more opportunity, one more thing through genetics that is being used in terms of heat tolerance, which is related to a specific gene. It's called the SLIC gene that was identified in other breeds, and they could trace back and have it for Holsteins. And there are a few countries that have used as well. And one thing that I think it's important to mention, considering this start in terms of implementing, was that we see, for example, Australia, it's a very important player in terms of the dairy industry and livestock in general, but also it has a different environment compared to Canada, and also a different system, right? It's a pasture-based system, and therefore, like Brazil, there are places where the environment may play a different role towards this, the animals, and therefore considering this environmental challenge. Now, when we think of this in Canada, this was actually the first part of my research here, was to really identify if Canada was really getting into troubles considering climate change and global warming, and the fact that it is a fact here in Canada. Even in the northern hemisphere, what happens is a few things that I can mention, but considering climate change, the northern hemisphere is actually being the one that is being more affected. So places, and we see in Canada, even in northern latitudes, the climate is changing very drastically, and the temperature is increasing, having a very hot summers and very cold winters. And considering then heat stress, it is a challenge. Also, the system, confined animals, so indoor animals, that may require more of the controlling of the environment. But even though, considering high producing animals, we've seen that the environment plays a very important role in how the animals is sensing and experiencing heat stress. So we looked in our, there is one paper published that we came up with numbers for a particular temperature humidity index value, that above that value, we could say for sure animals are experiencing heat stress unless there is some sort of alleviation in like heat abatement devices being implemented. And the numbers of days are quite impressive, considering a year, it goes over a month of animals experience or most likely experience heat stress. So I think that is sort of like how I could answer, giving a bit of information based on my studies. Again, countries that have implemented, they are looking into a short result, or not a short result, but an answer that is shorter because they have a different system. In Canada, we are looking at, because it will be something very important for producers even here. And we've seen in Germany the same situation, northern countries where temperature is also affecting production there. And that's another thing, considering Brazil, Australia, also those animals, they are acclimated. So there is that factor as well that needs to be taken into account. So among many things, I guess what we can say is that it is an issue that Canada needs to be looking at and to come up with ways to tackle this environmental challenge.
Thanks, Ivan. We are just at the top of our hour here, Saeed. There have been some questions that have come in for you, so I will share them with you by email, and we will get responses back out to the participants. Unfortunately, we are at the end of our time, and I see our participants dropping off here. So I just want to say thank you so much to our presenters today. Thank you to everybody who has been a participant in our session. It's been great to have you. As I mentioned, this is our 12th and final session. So for those who've been participating in the seminar for the last 10 months, thank you so much for joining us every 3 weeks. I also want to thank DFC, Dairy Farmers of Canada, for their support in putting these webinars together, and the sponsors that Amy has thanked at the beginning of our session today. And lastly, thank you to our presenters and all of our participants. Everyone, please stay tuned to the ACER newsletter and social media pages. We have lots more to share for the Cluster 3 and IRC projects as this year unfolds. So please follow along with us there. And we are looking very forward to seeing you all in the future. So thank you so much. It's been a pleasure. Thank you to our speakers today, and we will see you all again soon.